review quiz 4, the answers to quiz 4. Make sure that everybody understands what the answers to those questions are for your benefit and in case they might come up again. Okay, everybody see quiz 4?
Yes.
Make it bigger, okay. The first question, match the polymer with the monomer. And it came from this slide, which shows monomers on the left, corresponding polymers on the right. Nucleotide is a monomer for nucleic acids. Amino acids are monomers for proteins. Monosaccharides, single sugars, are monomers for polysaccharides, and fatty acids are monomers for complex lipids. Second question, the list below indicate the substance that is not a type of lipid. Well, one of the slides we looked at listed types of lipids. 5 different types of lipids, and we talked about each one of those. And on this list, they're all there except for spider silk, which is a protein. The third one is true or false. DNA and RNA molecules are constructed from nucleotides, each of which contains a phosphate, 5-carbon sugar, and a nitrogenous base. And that is true. Shown on this slide that we had previously. Nucleotide chain of monomers makes a nucleic acid, and each monomer, each nucleotide has those 5 units. So that's a true statement. Which of the following is not a difference between DNA and RNA? Well, the first one is not a not difference. So they're single-stranded versus double-stranded. The second one is not a difference. Covalent bonds versus ionic bonds. Both DNA and RNA are chains held together by covalent bonds. So that is not a difference between them. Taken from this slide. The next one is true or false. All amino acids have 2 functional groups attached to a central carbon. One is a carboxyl group, the other is a phosphate group. Here's where that question came from. All amino acids have these 2 functional groups in common. One is an amine and the other is a carboxyl. A carboxyl is one and an amine, not a phosphate group, but an amine group. Then I showed you a drawing of a dipeptide and asked you to tell me the letter pointing to the bond, which is a peptide bond. And most of you got this right. It's B. B is the peptide bond. In the context of a C double bond O and H. And that was illustrated in lecture by this slide. Peptide bond. Incomplete proteins are those missing 1 or more essential amino acids. Proteins of some plants are incomplete. And I've listed a bunch of plants and what they're missing. And the question asks to indicate 2 plants which together would provide all the essential amino acids. And many of you said rice and beans together, and that's correct. Rice is missing lysine, but beans have it. Beans are missing methionine and tryptophan, but rice has those. So you can have a complete balanced diet eating those 2. Now, there are other combinations that would work here, and I accepted any combination as long as they ended up complementing each other.
Professor, I got a question. This is Taylor.
Uh-huh.
So what I put for that answer was soy and rice. So is soy just considered a bean?
Yeah, soybeans are beans. I don't know the nutritional value of soybeans. I thought that they might be a complete protein in and of themselves. I don't know. Okay, if you equated soybeans with beans, then I'll give you credit for that. I'll mark that down. Did you lose a point on that?
No, I didn't. You gave me credit for it. I was just wanting to clarify.
Oh, okay.
It was just in the bean category.
Yeah, at the very least, they're both legumes. You know, all beans are legumes, including soybeans. Okay. Yeah, you just reminded me, Ben, that when you speak up, give me your name so that I can record you as having participated. Next question, what is a gene? And here's the slide that states what a gene is. Section of DNA that codes for another molecule, usually a protein. And quite a few of you had this down exactly as stated here. And finally, question 9. Isomers are 2 compounds with the same what, but different what? Well, on this slide, it's stated explicitly, isomers have the same chemical formula, so that was the first 2 words, but different structures. That's the last blank. Now, I gave quite a lot of latitude here for words that meant the same, but they weren't exactly what the definition calls for. For example, instead of structures, some of you said different arrangements. Fine, I accepted that. And the last one, a monounsaturated fatty acid differs from a saturated fatty acid by having a single double bond in the chain, shown on this slide from the previous lecture. Monounsaturated is a chain that's fully saturated, except at one point there's a double bond. So monounsaturated means there's one double bond in the chain. Anything more than one would be called polyunsaturated. So you could have 2 or 3 or 4. And you may notice when you read labels on food, often it will say polyunsaturated fatty acids or polyunsaturated components in the food that you're buying. And that's because polyunsaturated foods are easier to digest than saturated ones. Okay, I just wanted to run over those questions and answers quickly. Any questions about anything? One of the bad things about having flu symptoms is you have to drink a lot. So that's what I'm trying to do.
Hey, Professor.
Yeah.
When I got my second shot, they told me to take Tylenol if you have a fever and it might help.
Yeah. And I did.
Okay.
Did you have a bad reaction to your second shot?
I got extremely lucky and didn't feel a thing.
You are the one lucky person.
Yeah.
My first shot, I felt nothing. Absolutely no reaction, either at the injection site or elsewhere. But within a few hours after the second shot, I got hit really hard. I was in bed for the whole next day.
Yeah, everybody at my work had to call out the next day or were extremely ill, vomiting, all that.
Yeah, everything you expect to get in the flu, you get it with this reaction. And it's not really the virus that's causing it, it's the adjuvant that Moderna puts in it. People who get the Pfizer injection don't get that.
Well, I had Pfizer, so.
That explains that. Okay.
Professor, I got a question on that.
Uh-huh.
Because I'm getting my first shot on Thursday, and then the second one when I'm supposed to. But did you feel any respiratory problems? I get chronic bronchitis really bad, and that's scaring me from what people are telling me.
No, I didn't have any respiratory problems, although my blood oxygen level went down to 80%.
Oh, wow.
And so I was a little nervous about that, but now it's up to normal. So, but no, I don't know anything about effects on bronchial condition.
All right, thank you.
Okay, now I want to get into our topic for today, which is mutations. Mutations and the CFTR protein. CFTR is for the cystic fibrosis transmembrane regulator protein. And you may recall that I have from time to time mentioned that you will have a cystic fibrosis project in this course, and you will. And this is the start of the introduction to that. We'll get into a serious explanation of it next time. But for now, we're taking this protein as an example of a mutation. This slide's okay, so what is the CFTR protein? Well, it's encoded by the CFTR gene. Where is it located? The location code is given here. Does anybody remember how to translate this code? 7Q3-2.1. That's the location of the CFTR gene.
I remember what the 7 means.
What's the 7?
The 7 means the 7th chromosome.
Okay, and you're Katrina.
Katrina Vibert.
Okay. 7th chromosome, right. So what does Q mean? Anybody?
A Q mean a long arm.
Q means the long arm. That's right. What about 3? Well, 3 means the region of the chromosome. It just designates a fairly large region. -2 means the band within region 3. And 0.1 means it's the first subband in region 3. So that's how you read a code, and you should know that just in case it comes up again. Now, how many nucleotides make up this gene? About 250,000. So that's a lot. And those 250,000 nucleotides code for 1,480 amino acids that fold into a protein. Now that's many more base pairs than those that required for 1,480 amino acids. Now, how to explain this? Well, the answer is introns, primarily introns. And here's a video that we won't take time to go into, but you can look at this and you can see just how big this gene is. And it's not atypically big. Many genes are huge like this. And the normal function of CFTR is to transport chloride ions across the plasma membrane. So what are introns? This slide shows exons as wide gray bands and introns as narrow gray bands. CFTR has 27 exons and 26 introns. You can see them all lined up in a row. And the reason there's always one more exon in any gene than there are introns is because the gene has to start at some point, and it starts in an exon. And it has to end at some point, and it must end in an exon. So there's always one more exon than introns. So why? What's the significance of introns? Because they're spliced out. Intron splicing in messenger RNA. If you look to the left, you can see a DNA strand with the exons in blue, the introns in red. They're transcribed to a pre-mRNA with the introns and exons all left in. But then when it's translated to mRNA, the introns are spliced out. So you end up with this bar that's only blue. All the introns are gone. It's only the exons stitched together. And these then get translated into proteins, which go on to acquire a tertiary structure, the form in which they are active. Now, the things to remember about this are that intron splicing occurs only in the nucleus. And intron splicing occurs only in eukaryotes. Prokaryotes don't have it. So it's clearly not essential for life, but it's found in virtually all eukaryotes. Why? Why are there introns? Well, some of them are ribozymes, which I'll mention in a minute. Some are regulatory sequences, which determine when real genes turn on and off. And then there are other functions for introns which are undiscovered. So now we have an additional type of RNA. You know, from the past, we have mRNA, tRNA, and rRNA. Add to that now ribozymes, which are catalytic RNA. So the name comes from ribonucleic acid enzyme. Abbreviate that to ribozyme. And what ribozymes can do is hydrolyze phosphodiester bonds in a nucleotide chain. So the bond between the sugar and the phosphate can get cleaved by a ribozyme, cutting the chain in two. You see that illustrated here with an RNA message in blue a ribozyme sitting on a recognition site. And if it cleaves that, you get 2 pieces. And it will cleave at the same site every time so that all the pieces, the short pieces are the same and all the long pieces are the same. So that's one thing that ribozymes can do. Here's an example of how a ribozyme can regulate the expression of a gene. If you have a gene here in yellow, the ribozyme can sit on the mRNA. If you inhibit that ribozyme with some molecule, it can't function. It can't damage the RNA and the gene is expressed. If, on the other hand, the ribozyme is not inhibited, it's active and it cuts the RNA and that gene is not expressed. So that's like a switch. If the ribozyme is active, it turns the gene off. If it's not active, the gene's expressed. Now, introns have options called alternative splicing. And it's another means of regulating gene expression and also introducing variation into the genome. So you can have full splicing. What you have here are 3 exons in red, blue, and green, and 2 introns. Well, if both of the introns are spliced out, then you have exons 1, 2, and 3 joined together. Or you can skip both, skip the exon, skip 1 exon, in which case it's eliminated and only exons 1 and 3 are joined. Or you can have alternative 5' splice selection, in which only part of exon 2 is present, or the alternative 3', in which the other end of exon 2. And you can have intron retention, in which one intron is left in and becomes part of the gene that is translated. So you can see how by differential splicing of introns, you can get several different configurations of a gene.
Professor,
Yeah.
this is Leanne Metzger. So with that, even with those alternative splicings, those are still the ribozymes splicing them, or the alternative is something else? I didn't understand. I'm sorry, I didn't understand.
Your question was, does the intron that's left there, does that become part of the gene?
No, so the ribozymes are still the ones doing the splicing, right? The alternative intron splicing.
Oh, no, ribozymes are not involved in splicing out introns.
Okay, that's where I got confused. Okay.
Yeah, okay, that's good to clarify that. Ribozymes just have another function for regulating genes. Okay, so this is the CFTR protein, a diagram of it, and you can see it's embedded in the plasma membrane, the phospholipid bilayer. And it extends from inside the cell interior to outside the cell exterior. And its function is to transport these chloride ions, these balls, through this channel to the outside of the cell. And this is critical for the function of the organ, which we'll come to later. And so the protein is folded we talked about bonds that cause folding, so that it ends up as sort of like a silo, a circle, which makes a channel. And it's that channel that chloride ions travel through. And so there are two transmembrane domains, 1 and 2, in the two different colors. There is a regulatory domain, which determines the regulation of the channel. And then there are two NBDs, nucleotide binding domains. So what are they and what are they for? Okay, nucleotide binding domains. You remember what nucleotides are? There are 4 of them in DNA, there are 4 in RNA, and this shows adenine in its triphosphate form, ATP. And you may be familiar with the term ATP. It's the so-called energy currency of the cell. So cells derive energy from ATP. How do they do that? Well, this last bond in the triphosphate is a high energy bond. So when it is hydrolyzed, water goes in, a phosphate ion comes out, and so does energy. And you end up with ADP, adenosine diphosphate. So when it refers to a nucleotide binding domain, it's referring to the adenine base being hydrolyzed to the diphosphate form with the release of energy. And what is that energy used for? I need to back up. The energy that's released by these nucleotide binding domains is used to transport the chloride ions. The transport is active. It requires energy. The chloride ions don't just flow through this channel by themselves. They have to be transported with energy. So the hydrolysis, the energy is used to change the configuration of this channel so that the chloride ions can get through.
And then Professor, can you just tell me what ADP is? Like,
ADP is adenosine triphosphate.
and what does that exactly mean? Like, what does that translate to? It like how
Well,
ATP is energy?
Yeah, ADP is not an energy currency. ATP is because it has this third phosphate attached in a high energy bond. So when ATP is hydrolyzed, you get ADP plus energy. And then ADP can be rephosphorylated back to ATP. It can keep getting cycled. And of course, that rephosphorylation also takes energy.
Okay, thank you.
Okay, let's go through the route that the CFTR protein travels on its way from being encoded in DNA in the nucleus until it finally ends up in the plasma membrane over here on the right. So it's encoded on chromosome 7, and it's transcribed and translated into mRNA. And remember, this all happens in the nucleus. But the mRNA can actually leave the nucleus through a pore. And when it does, right outside that pore is the endoplasmic reticulum, ER. The endoplasmic reticulum is where the ribosomes are. So here's where mRNA, tRNA, and ribosomal RNA get together in order to make a polypeptide. Well, once that's produced, remember this is a primary structure, it gets transported to the Golgi apparatus where it is decorated, so-called decorated. That means sugars are attached in a process called glycosylation. The sugars are necessary. In addition to folding, they're necessary for activity. And then this decorated protein gets transferred into a vesicle. The vesicle can fuse to the plasma membrane, and in doing so, it deposits the CFTR protein in the plasma membrane, making a channel through the membrane. So that's the route that the protein takes. And what we're going to talk about is how different mutations in the gene will block this route, this course at various different places. This slide on the previous slide, there was a link to a video, which I'd encourage you to watch. I don't want to take time to do it now, but in the video, a guy does a drawing of that trip that the CFTR protein takes from the nucleus to the plasma membrane. And he's illustrated various steps of that, which I've taken screenshots. So the pink bars are exons, and between them are introns, get spliced out, you get this mature mRNA. And he talks about how that gets transferred to ribosomes, and the protein, the polypeptide is made, which needs to be decorated. So a major part of what he talks about is this decoration where sugars get added in the Golgi, and ultimately after the sugars are added, it gets put into the plasma membrane. And he talks a lot about core sugars and complex sugars. The core sugars are single chains unbranched of monosaccharides. The complex sugars are branched. And this just illustrates a core sugar, just alpha-1,4 bonds between 3 molecules of glucose. The glucose chains can be much longer than that, of course, but they're unbranched. The core sugars are highly branched. So now we're going to switch away from specifically the CFTR protein and talk about mutations in general. What is a mutation? It's any change in the nucleotide sequence of DNA. So remember that. It may result in a change in the amino acid sequence, or it may not, and we'll talk about that in a little bit. Now, if the mutation does cause a change, the protein may not function properly. Mutations are the basis for permanent variation in organisms. There are other mechanisms which we'll talk about when we deal with cell division, other ways of introducing variation, but they are not necessarily permanent. Mutations are. They're the raw material for natural selection to choose the fittest. And mutations can cause genetic diseases, for example, cystic fibrosis. Cystic fibrosis is not contagious. The only way it can be caused is through mutations in the CFTR gene. Now, what causes mutations? There are a number of different ways that a mutation can occur. One is simply by replicating the DNA. You remember last time we talked about the 6 steps in DNA replication, the enzymes involved. You remember that replication is really done by DNA polymerase 3, but it doesn't do it alone. There are several other enzymes that are required to help. So it's a very complicated process. And what causes mutations? It's a complicated process, and because it is so complicated, mistakes can happen. And so just by the normal replication of DNA, we can get mutations. There are certain chemicals that damage DNA, such as intercalating agents. These are small molecules that insert themselves into the nucleotide chain. And there are carcinogens. And there's radiation damage to DNA. If we go out in the sunlight, where we get a dose of UV radiation, that can cause mutations. And of course, ionizing radiation is even more mutagenic. But finally, what can cause mutations is genome editing. Now, this is the hot topic in biology today. It's literally revolutionizing how biology is done. It's a process called CRISPR, and I hope to get to that later in this session and describe to you what CRISPR is and how it works and why you should know about it. Okay, here's an example of how a mutation can occur. So if you look in the upper left here, you see a DNA chain, and one of the 4 bases in DNA is thymine. If there are 2 thymines in proximity to each other, they don't need to be adjacent, but they have to be in the same neighborhood. And if that piece of DNA is struck by ultraviolet radiation, what can happen is a pyrimidine dimer. You see that here, where two new bonds have formed, linking these thymine residues. And it's important to note that these bonds are covalent. So remember, what's holding the 2 chains together, these are all hydrogen bonds. But in a pyrimidine dimer, they're covalent bonds, very strong. So what is a pyrimidine? Well, we have to reclassify the 4 nitrogenous bases. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. Pyrimidines do not bond to themselves, and purines do not bond to themselves, but they do bond to each other, hydrogen bond. So T and A, hydrogen bond, a pyrimidine and a purine. 2 hydrogen bonds. Cytosine and guanine can bond with 3 hydrogen bonds. Again, a pyrimidine bonding to a purine. So thymine, being a pyrimidine, it's why it's called a pyrimidine dimer. So this illustration here on the right shows a DNA backbone with 2 thymine residues sticking out. When they're hit by UV light, thymine dimers occur. And as I said, these red bars are covalent bonds, so they're very firm and they remain. And now imagine DNA polymerase 3 comes along copying this DNA. So remember, helicase unwinds the DNA so that all the enzymes can get into the space and do their copying. And the thymine dimer causes a kink in the backbone so that when DNA polymerase 3 goes through here, copying the bases, it makes a mistake because it can't deal with this kink. And that mistake is a mutation.
Professor, at
Yeah.
that point where it breaks, are they back to RNA?
No, this is all DNA.
But when it breaks, there's nothing holding it together in the double helix. So they're single stranded apart.
Well, remember, after the enzymes pass through, the chains go back together. So
Okay,
it's then
gotcha.
a double-stranded DNA.
So once it passes the mutation, then
Yeah,
it goes
after-
back to normal.
Right, after it passes the thymine dimer, it's back to normal copying, leaving the mistake behind it. The copying error is left, but that then becomes a permanent part of that DNA. So let's take a couple of examples of how mutation works. Here's an example, an original DNA strand. And in the yellow, you see the sequence, TACAGT, and so on. And then supposing a mutation occurs so that this T in the original strand becomes an A in the mutant. So what is the effect of that mutation on the peptide? Well, to answer that question, you first transcribe the DNA, the mutant DNA, to messenger RNA, and then you translate it to protein using a codon table, which you see here. So when you study this material, you should run through this exercise yourself by hand so that it sticks in your mind. So that is example number 1. Now, example number 2 is the original DNA strand. And now the mutation, instead of a G here, is an A. What is the effect of that mutation on the peptide? And again, you translate that to mRNA, you look up in a codon table, and you find out what the effect is on the peptide. Now, I'll show you a summary of the answer, and you can check your result with this. But we look at the 3, the normal sequence, mutant 1 sequence, and mutant 2 sequence. In the normal sequence, if you translate mRNA to protein, it's MET, SER, asparagine, cysteine, ARG, and stop. Now, if we look at mutant 1 sequence, where this A now becomes a U, instead of AGA, AGA is the codon for serine. And if we look up UCU, it's still serine. So the peptide sequence didn't change. Even though we have a mutation in the DNA, the peptide sequence remains the same. So this is called a silent mutation. It is a mutation, but it has no effect. Now consider mutant 2, where this G becomes an A. So now instead of the codon AAT, it's UUA. And if we look that up, UUA is leucine. So now instead of having a peptide that is met ser ASN, it's met Lu ASN. So that's a real mutation because it caused a change. I shouldn't say real mutation. They're all real mutations. This is a mutation with consequence because it changed the sequence of amino acids. And since it's a single base pair caused it, it's called a missense mutation. So remember that. Now, this is a summary of what types of mutations do we have? I just pointed out what a point mutation is. It's one base pair change. And all the mutations in this table, which we'll come to in a bit, are point mutations. But that's not the only kind of mutation that you'll find. In fact, other kinds of mutations are equally common. And they're listed here. They're self-explanatory, and you should remember these. Remember what a duplication is. It's a piece of DNA that gets copied and then repeated. It stays in the genome and it gets repeated. It can be repeated many times, over and over. That is a duplication. It can be a deletion where a piece of DNA is simply removed. An inversion is just that. A piece of DNA is flipped over, so now the sequence reads backwards, and you get a completely different peptide out of that. An insertion is a new piece of DNA is introduced into the sequence. And finally, a translocation is a bit more complicated, where two pieces of DNA exchange places. Now, this is the most common type of translocation involving two pieces of DNA. However, some translocations involve just one, where one sequence jumps from one place in the genome to another without a reciprocal response. Now, what are the effects of point mutations on polypeptides? Well, as we've just seen, a silent mutation has no effect. It's a mutation, but it doesn't change anything. A missense mutation is a one base pair change that does alter the protein. A frame shift shifts the entire frame of the DNA sequence so that anything after a frame shift mutation is different from the wild type. And finally, a nonsense mutation is stop. If there's a stop codon in the protein coding region, it's called nonsense. And it means that the polypeptide terminates at that point, and it's very likely to be inactive because it's terminated early. And now the table on the right here is simply to illustrate these various different types of point mutations. So the first line is a wild type. The second line shows a silent mutation where this G becomes a C, doesn't change anything. Lysine is still lysine. The third is a missense mutation where this G becomes a C and it changes alanine to proline. The next is a nonsense mutation where there's where this A becomes a T and introduces a stop codon. So the polypeptide stops at this point, and what's left is probably inactive. And finally, a frame shift, or the mutation is this A. Everything after the point of the frame shift is different, all different amino acids. And again, it's probably such a drastic change that the protein is inactive. Okay, so this is an important slide. You should know all the different types of mutations and what their effects are.
Professor,
Yeah,
this is Anna Mendez. I just have a quick question. I'm confused on the myces. Sorry, the misins. Could you give an example, more of a clear example of that?
of what?
Of the misins?
The missense.
Sorry, missense, sorry.
Okay, a missense mutation is, first of all, a point mutation. So you can see that here. This G becomes a C. So there's a single base pair change. And what's the effect of that? The second amino acid is normally an alanine, and now it's a proline. So a missense mutation is a single base pair change that results in a change in amino acid. And that's in contrast to a silent mutation where you have a mutation, but it doesn't change anything. So in the case of the second line down here, this C, changing this G to a C, does not change the amino acid sequence. The wild type is the same as the one with the mutation. So that's a point mutation that's silent. A missense mutation is a point mutation that is not silent. It changes the amino acid sequence.
Got a question.
Uh-huh.
So I'm also taking physical anthropology, and we just studied the forces of nature with their subgroups. And one of the things that we were talking about in a video we watched was sickle cell, which was a point mutation. But I never found out which one it is. Does that happen in frame shift with the addition of that misshapen blood cell?
I don't know. I do know that you're right. Sickle cell is a point mutation. So there's one amino acid change, and that one amino acid, it's in hemoglobin. It's in the hemoglobin gene. And that one amino acid change affects the ability of hemoglobin to carry oxygen. So
I'm pretty sure it's frame shift because it inserts that.
I don't think it's frame shift. Frame shift would change the entire amino acid sequence. And that's not the case. In the case of sickle cell, it's a hemoglobin molecule with one amino acid difference. And that one amino acid change alters its ability to carry oxygen. And so that's why sickle cell patients have trouble getting enough oxygen.
So would that be missense then?
That would be a missense mutation.
Okay, thank you.
Okay, anything else on this? This is important. Okay, let's talk more about CFTR mutations. Over 2,500 are known in this one gene alone. Most of them are single base substitutions or deletions. The most common mutation is called F508del. That means that the phenylalanine residue at position 508 is deleted. And this mutation is present in about 70% of the people who have the disease. Now, you have this large number, 2,500 mutations that are possible, Most of them do not result in symptoms. Only about 400 of the 2,500 actually cause disease. And finally, about 1 in every 31 Americans carries a mutated cystic fibrosis gene. However, you have to have 2, 1 from each parent, in order to have the disease. So it's only if 2 carriers have an offspring, does that offspring have the disease? And then the symptoms can be highly variable, and depending on what the mutation is. And this is where your project for the semester comes in. You will each be assigned a mutation, and you will research that mutation and report to the class at the end of the semester what you find out. And I'll tell you next time what the components of your project will include. No. This shows the spatial locations of mutations with del F508 as an example. If the same mutation occurs on each of your chromosomes, remember you have to have, if this chromosome is from your father and this one from your mother, they both have to have the mutation, F508. If it's at the same position, then it's called a homozygote. Now, it doesn't have to be 2 of the same thing. So one chromosome can have a delta F508 mutation, and the other chromosome can have a different mutation in the same gene. And that can also cause disease, and that's called a heterozygote. Or both mutations, deletion F508 can occur at one position, and there can be another cystic fibrosis mutation at a different position on the same chromosome. And those are called mutations in cis, as opposed to this other one, which are mutations in trans. So now let's talk about the different classes of mutations, and you will be assigned one of these 6 classes. So if you start in the nucleus, the mutation can cause a copying error in DNA, so you may never get a transcript, or the mutation may occur in the transcript so that you get a defective protein that never does go any farther than the endoplasmic reticulum. Then there are some mutations that affect the decoration, the attachment of sugars to the protein. There are other mutations that don't allow complete transport to the membrane. And then there are other mutations where the protein gets all the way to the membrane, gets inserted in the membrane, but it doesn't work right. So it can be rare, it can be fewer than normal, it can be inactive, or it simply works less efficiently. But the result of all 3 of these, even though there's protein in the membrane, is that not enough chloride ion is transported. And if there's not enough chloride ion, then mucus builds up, and we'll talk about the effects that this mucus can have on organs next time. This illustrates those 6 types of mutations in a graphic form, starting with normal, no mutation at all, 1 through 6. So you see that class 1 and class 2, no protein ever gets to the membrane. So they're null mutations. Patients that have this type of mutation have very serious problems with cystic fibrosis, with clogging up with mucus. Class 3, you get a protein in the membrane, but the channel is blocked, so nothing goes through it. Channels 4, 5, and 6, in every case, the protein is in the membrane, but it's defective for different reasons, resulting in less chloride ion transport than normal. And there's enough of a reduction to cause disease symptoms. So as I said, the different types of mutations here, the names of the mutations for each one of these classes, and you will be assigned probably one of these. But whichever one it is, you'll need to do research on it to determine what's wrong with the protein. Is it a point mutation? Is it deletion? Is it insertion? What is the effect of that mutation on the function of the protein? And that will be up here in this top row. This is the function. So what is the effect of your mutation on function? And finally, what treatments are available to treat patients that have your mutation?
Professor, I have a quick question. Is that going to be a group project or an individual?
Yes, it will be. I'm not sure how the groups will be assigned yet, but they will be. So each group then can decide how to do the research, how to put together a presentation, and then who's going to present it as a PowerPoint presentation to the class.
Okay, got it. Thank you.
This is a group assignment?
Yeah, the groups will be small. I imagine 3 or at most 4 people per group.
Will they be randomly assigned or do we choose our groups?
Haven't decided yet. I'll get to
Okay.
that next time. I'm running out of time. I won't go through this table in detail. It simply shows you the nomenclature for mutations, and in particular CFTR mutations. So if you look at the first one, 254G goes to A, that means that base 254, where normally there's a G, has been mutated to A. And that means that glycine is now glutamine. And it's a missense mutation because it's a point mutation that changes the amino acid.
I had a question. It's Justin Hansen.
Okay.
So the only way to know if the glycine is changed to the glutamine or whatever you said, that's on that table that you showed earlier, right? That's like the only way.
The codon, yeah, the codon usage table. That's how you figure out what the change is.
Okay, thank you.
This slide. Now I made a summary here, summary points for CFTR and mutations. I'm not going to go through this now, I don't have time, but this is for like a study guide for you to read these questions and answer them. And it should help you in studying for tests and quizzes. And now I promised you CRISPR. I'll go through this rather quickly. Last year, 2020, the Nobel Prize in Chemistry was awarded for CRISPR. The recipients were Jennifer Doudna, who was a professor at UC Berkeley, and her colleague from France, Manuel Carpentier. Together, they worked out the details of how to do a very special kind of mutagenesis, which is extremely valuable. And you'll see why in just a minute. So how to cause mutations, the old way versus the new way. Well, you can wait for a mutation simply because you know that because of replication errors, they will eventually come. But you don't live long enough to get mutations in that way. They occur too infrequently. Radiation and chemicals can damage DNA and cause mutations, but what's the problem here? If you damage DNA with, say, UV radiation, you're damaging not necessarily just the gene you want to mutate, you're damaging the entire genome. So you have mutations all over the place, and you have to sort through them to find the one you want. And that's where CRISPR comes to the rescue. Genome editing is what it's called. And here's what causes mutations. Here's how it works. First, before I get to that, if you, as I just said, if you wait for a naturally occurring copying error, it's too inefficient, you'll never find it. And mutagenesis of all different kinds is nonspecific. You mutagenize the entire genome, whereas CRISPR, look at this. Why is it so important? Because it permits any mutation. That means any gene in the genome can occur, you can induce it in any organism, from prokaryotes to eukaryotes. Any organism it's been tried in, it works. So here's what you remember. CRISPR is important because it is site-specific, means it targets the gene that you want to target, and it's universal. You can do it in any organism, no matter what organism you're working with. And this little diagram shows, it's supposed to illustrate the precision, where you take forceps and you just pluck out the little piece of DNA that you want to remove. Okay, how does it work? It's remarkably simple, considering how complex the system is that it's derived from. For gene editing, well, first of all, I need to point out that Cas9 is the enzyme that's critical for CRISPR, because it cuts the foreign DNA at a specific point. So if you want to do CRISPR yourself, you need two things. One, you need a guide RNA, which you synthesize in the lab. It's a short piece of RNA, maybe 15 or 20 nucleotides long, which is easily made artificially in a test tube. And you need Cas9, which is a very special enzyme. It's an endonuclease. So the way this works is you make your guide RNA, you synthesize it, and you mix it with Cas9 protein, and it makes a complex. Then you add that complex to genomic DNA of your favorite organism. That happens to be humans, and you add Cas9 guide to human DNA. And you've constructed your RNA guide to correspond to the gene you want to mutate. So the gene you want to mutate is here in red. The guide is in purple. So since this is the complement to your target gene, it matches up perfectly. Then if there is a spacer called PAM at the end, Cas9 will cut the DNA precisely, both strands. So you end up with DNA with a gap. And this gap then is useful in that you can just close it up, and that itself is a mutation. You've caused if a piece is deleted, you've caused a deletion, or you can put in any sequence that you want. So for example, supposing your target was a mutated gene, and you made a sequence without the mutation, you made the wild type sequence, you could precisely cut out the mutant segment and put in the wild type segment. So I think you can see why this technology is so powerful and why it got the Nobel Prize.
Is it still considered unethical?
only if you're doing something unethical. It's like any technology, it can be used for good or evil. So when the technology was first described, there was a group in China that used it on human embryos. And that was roundly discouraged throughout the world. Everyone said, you don't do this with humans. But it's been done with many other different kinds of organisms that people do not consider to be unethical manipulations. It's really no different from any other type of mutagenesis, except for its precision. So in any other type of mutagenesis, you get things you don't want. With CRISPR, you only get what you targeted. And that's really part of the power of it. That and the fact that it's universal. It works on any organism.
So at this point in time, CRISPR is not really in a human application, correct?
That's right.
Okay.
Yeah, it's considered unethical to do that. And I don't
Professor?
know.
Oh, sorry. Go.
Well-
Kiam Anderson. I just had a question. Since what would be like an ethical use of CRISPR?
An ethical use?
Yeah. Like what is
Well-
something that we use it on that's
Well, take, for example, the cystic fibrosis. You wouldn't do this in an embryo, and so it wouldn't be a permanent genetic effect. But supposing you had a point mutation in the CFTR gene that's causing cystic fibrosis. Well, if you just pluck that mutagenized, or the mutant region of the gene out, replace it with wild type, and put it back in the cells, now you've got a cell that is no longer subject to cystic fibrosis. The big problem in doing this is it's fairly easy to do it in cells, but how do you translate that, or how do you extend that to all the cells in the organism after the organism is already formed? Because remember I said you can't do this in embryos. So it's fairly easy to do what would be considered ethical things like cure a disease. It's fairly easy to do it in a cell. It's not so easy to do it in a whole organism.
Does it become unethical because clearly you'd want to prevent diseases or make people immune to, stuff like that? Is it unethical because it kind of makes it like class, like rich versus poor?
No, it's unethical because it's a change in the germline.
Okay.
If you do it in an embryo, all the cells that develop from that embryo into the body of the individual, they're all mutant. And so people are concerned about designer babies. If genes, say, for intelligence could be identified, then if you were genetically manipulating embryos, you could try to get those genes concentrated in individual embryos.
So Professor, like in China, when they clone those two macaque monkeys, is that where it's considered unethical?
Depends on who you talk to. Most people don't consider doing it in monkeys to be unethical. It only becomes unethical in humans. But that itself is controversial. Not everybody would agree with what I said.
So it's still up for debate.
Yeah, and to be honest, CRISPR is being used in animals of all kinds, and plants, and microbes. This slide shows on the left definitions of some terms that you should know, and then it's another illustration of how CRISPR is done. The two critical components are the guide RNA and Cas9, the enzyme. They form a complex. Here's your genomic DNA in gray. The guide targets the gene of interest, And if you have a spacer called PAM, Cas9 cleaves the DNA, both strands. And once this happens, you can get any different kind of mutant, an insertion mutant, a deletion, or a frame shift. So again, I'm repeating what I'm saying because it is so important. CRISPR has extreme target specificity. So you don't have to worry. You do, if you make a mutation with CRISPR, you still check the genome to make sure that you haven't caused damage elsewhere. But in general, it's very specific for just the target that you have chosen. And it works in any organism. So the specificity and the universality are the two things that are unique to CRISPR. There's no other mutagenesis that it has these two features. So it works on viruses. You can imagine destroying viruses, including coronavirus, using CRISPR. And I know there are pharmaceutical companies working on that. I don't know that there's been any success yet. But you could, in principle, correct genetic flaws, like in cystic fibrosis and muscular dystrophy, or sickle cell anemia. It can be used in crops, in ecosystems, endangered species, and so on. There's almost nothing that you can imagine that CRISPR, in terms of biological variation, that CRISPR won't do. Why you should be knowledgeable about CRISPR. It's revolutionizing biology. It shows up frequently in news reports almost any day. If you go through the news, you'll find reference to CRISPR being used on yet something else. So for that reason, I think it's important that you know what CRISPR is, what it can do, and what its limitations are. Okay, I think, in fact, I know because I've gone over time, I'm sorry. I'm going to stop sharing. And if there are any questions, go ahead. Otherwise, I'm sorry I took up too much time.
Thank you, Professor.
Yeah, thank you. Feel better.
Yeah, feel better.
I hope I'm better on Wednesday. Okay, I'll see you then. Remember, there'll be a quiz, and we'll talk about cystic fibrosis some more. Okay.
Okay,
Take care.
thank you. Bye.
Bye.